This is the microarray, so DNA microarray slide. So here you are differentiating the muscle cells with the pancreatic cells. So this DNA microarray contains a holes, and those holes will fill with the IBTA. So the DNA stands. So these are all the steps that are involved in the DNA microarray technique. So sequence the entire genome organism, use massive amounts of computing power to figure out where all the genes are in your sequence. So do more computer crunching to design primer pairs. So using robots, run quality control test, repeat the steps with the 3 and 5, separate double-stranded DNA, use robot to place microscopic droplets, use more computers to create and manage a database, do more quality control to make all the stuff parts contain equal amount of DNA. These are the steps that are involved in the DNA microarray. So here also showing the sample, the DNA microarray slide. So these are all the holes that are in the DNA microarray. Each hole contains a single stranded DNA. So here they are doing some DNA microarray test for the healthy cells with the cancer cells. So this is the healthy cells, this is the cancer cell. So the gene 3 is off in the cancer cells. So that is switch on in the normal cells. Because of gene 3 switch off, so there is a mass of cancerous cells. So this gene 3 that is switched on in the normal cell. So these are the steps. So collection of tissue, isolate RNA, isolate mRNA, make labeled DNA copy, apply DNA, scan microarray and analyze data. So these are the 7 steps that are involved in the DNA microarray technique. So these are all the different solutions and the equipments that are needed for the microarray. Here we are showing this slide is showing the application of DNA. So DNA microarray helps especially in the identification of single nucleotide formal mutation, classification of tumors, identification of target genes in tumors, identification of cancer biomarkers, identification of genes associated with chromoresistance and drug discovery. So this is all the data, so that images will be collected and analyzed. So gridding, segmentation, intensity extraction and background correction. So these are the things that are done in the computers. So similarly translation into the clinical application, biological verification and interpretation. So data transformation and de-identification by using the images of the DNA microarray. So you can transform it into a some other map. So where the map is the collected microarray images are compared with the other images. So that are collected from different samples. So this is the method. And so simple examples, SNP microarray. So how it has to be applied in the patients, means patient specimens, abnormalities unknown and suspected to be small and unusual. So make the SNP microarray. So this is the method of, this is the application of microarray, and this is the application of microarray in classifying the cancers. So